Hello, I am Federico Luzzati, and this week I want to address with you the problem of the temporal patterning of neuronal progenitors in the development of the neocortex. In particular, I will focus on two recent papers that, using very similar techniques, arrive at opposing conclusions. And So the aims of this week will be thus not only to understand the problem of temporal patterning, but also to improve your capacity of critically reading scientific papers. And now let's start with some basic concepts. You should all know that the central nervous system is an extremely complex structure with an incredible cellular variability. As you can see from this single sagittal section, even a mouse brain can be subdivided into multiple areas, each containing its specific set of cell types. And so, a fundamental question in developmental neurobiology is to understand how this huge amount of neuronal variability is generated. The story of the development of the nervous system begins with an hollow tube, that is the neural tube, and that is delimited by a pseudo-stratified epithelium that is made by actively dividing neural stem cells. These neural stem cells are in contact with both the apical and basal sides of this epithelium and characteristically move their cell body towards the basal surface of the epithelium during the S phase of the cell cycle, while they come back to the apical side to undergo mitosis. At this early stages of development, these cells are believed to undergo mostly symmetric cell divisions that expand the stem cell pool. At the onset of neurogenesis, neural stem cells begins to divide asymmetrically, giving rise to daughter cells that leave the ventricular zone. These cells may also eventually divide again a few times before terminal differentiating, and in such case they are referred as intermediate progenitors, and these cells usually divide in a specific layer that is called the subventricular zone. But once become post-mitotic, newborn cells then migrate to their final destination, where they differentiate into mature neurons. In this slide you can see a more realistic picture of what's going on. These are images of coronal sections taken at different developmental stages and stained with the phosphohistone H3, that is a marker of mitotic cells. Here you can see that most mitotic figures are at the apical surface of the ventricular zone, and to a lesser extent also in this other layer that is the subventricular zone and that contain the intermediate progenitors. In this sequence of images, you can also appreciate the thickening of the brain parenchyma as a result of the accumulation of migrating and differentiating postmitotic neurons. In the past 20 years, we have learned that neural stem cells in different regions of the neuroepithelium are not equivalent. Rather, through various mechanisms of spatial patterning, the ventricular zone is regionalized and subdivided into multiple domains that show the expression of specific sets of transcription factors. The progenitors that occupy these different domains differ in their cell phase potential. This means that each ventricular zone domains give rise to a restricted repertoire of neurons that will then migrate to their final destination. As a further complication of this picture, the cell fate potential of ventricular zone progenitors do not only change in space, but also in time. Indeed, since the introduction of thymidine analogues in the 50s, it is well known that neurons are produced in definite sequence and in specific time windows. First neuron A, then neuron B, then C, and so on. Okay, and here you can see an animated version of this concept. And this depends on a sort of temporal patterning of neuronal progenitors. But how is this temporal patterning of ventricular zone progenitors regulated? As you should have already heard from Silvia, in Drosophila it is well demonstrated that a single neural stem cell or neuroblast can give rise to multiple cell time in a precise, ordered and tightly regulated sequence. In this species we are also beginning to understand the molecular mechanisms that regulate this temporal patterning. Unfortunately, however, the vertebrate picture is much less clear. Probably the vertebrate example in which the temporal patterning of neuronal progenitor has been more deeply investigated is the neocortex, that is a region of the telencephalon. And as you may remember, the telencephalon is divided into a pallium that lies dorsally and to a subpallium that is ventral. And the neocortex is one of the pallial regions, and it's specific to mammals. The neocortex is made by several areas that have different functional role, process different kind of information. However, despite their functional differences, all neocortical areas share the same functional unit, that is the neocortical column. A column can be divided into 6 layers, numbered 1 to 6 from the pial surface to the corpus callosum. As in other brain regions, in the neocortex we recognize 2 main neuronal categories that are the projection neurons, that are glutamatergic, in the case of the neocortex, and interneurons that are GABAergic. Projection neurons can be distinguished not only on the basis of their laminar position, but also because of their different connections. In example, upper layer neurons shown mostly intracortical projections, either callosal or not, while deeper layer neurons project to subcortical targets. Layer 5 neurons can have projection reaching the spinal cord, while layer 6 neurons predominantly target the thalamus. In the telencephalon of all tetrapods, multiple progenitor domains can be identified in the ventricular zone. Neocortical interneurons are generated in the subpallial ventricular zone, and in particular in this NKX 2.1 region, and from there they migrate tangentially to reach their final destination in the cortex. By contrast, projection neurons are generated in one of these 4 pallial domains, that is the dorsal pallium, and undergo a much more short trip, migrating radially. A very interesting feature of neocortical development that makes it an ideal model to study temporal patterning is that the 6 layers are generated sequentially from the deepest first to the upper layers last. In this image you can see the distribution of cells labeled by tritiated thymidine in the neocortex of adult cats. This analog was administered at different embryonic ages and then analyzed in adults. As you can see, if we inject tritiated thymidine at E50, we label only layer 6 neurons, while if we inject tritiated thymidine at E90, we label upper layer cells. When neurogenesis begins, ventricular zone cells, the neuronal progenitors, transforms into this characteristic cell type, that is the radial glial cells. These cells have their cell body, you see here, in the ventricular zone, and send a long process towards the pial surface. These radially oriented processes are used by immature neurons as a scaffold for their migration towards their differentiation site. It is now well demonstrated that radial glial cells are the primary progenitors of neurons and glial cells during development and in all regions of the central nervous system. This has been demonstrated primarily using a genetic lineage tracing analysis of cells that express the brain lipid binding protein, or BLBP. That is one of the main radial glial markers. Here I've mentioned also other markers, GLAST, nestin, vimentin, and so on. However, here the BLBP promoter was used, and in this kind of experiment the Cre recombinase is controlled by a specific promoter, BLBP in this case, and mediate the excision of a stop codon that ultimately leads to the expression of a reporter gene, in this case beta-galactosidase or beta-gal, in BLBP expressing cells and their progeny. In the adult brain of this mice line, virtually all new and positive mature neurons express also the reporter, the beta-gal, indicating that they originated from a BLBP positive radial glial progenitor. The role of radial glial cells as neuronal progenitors has also been demonstrated through in utero retroviral genetic lineage tracing, but now I will not go through a detailed description of this part here, as is not also directly related to the articles that you will have to study, but if you are interested, just download the presentation. And okay, so identifying the progenitors was a first fundamental step, but the next step will be to define the mechanism that regulate the temporal patterning in these cells. Historically, the prevailing model of neocortical neurogenesis is that projection neurons are generated sequentially from a common progenitor cells that, as in Drosophila, progressively restrict its cell fate potential. So it gives rise first to deeper layer neurons, then upper layer neurons, and ultimately to glial cells. Recently, however, a new model has been proposed in which different neuronal types are generated from distinct progenitors that segregates before the onset of neurogenesis and are then activated sequentially. So the hypothesis is that the occurrence of multiple progenitors, one for the glial cells, one for the upper layers, one for the deeper layers. And in this model, neuronal progenitors are a bit like comedians in a TV show. They are all in the theater before the beginning of the show, but they wait their time behind the curtains and then activates when their moment comes. Okay, the two papers that I want you to read this week are the one that proposed this new model of multiple progenitors, but also a subsequent one that reaffirmed, using similar techniques, the old theory of the common progenitor. Okay, but before showing you these two papers, I will describe you briefly two main experimental strategies that have been used to support this first model of the common progenitors in cortical neurogenesis. The first approach has been that of heterochronic transplantation of ventricular zone progenitors in the ferret, that is this nice animal here. As you can see from this birth dating experiment, if you give tritietethimidine at E36 in the ferret, you obtain cells that are mostly in layer 4 and to a lesser extent in layer 2-3. But if you transplant, if you isolate these ventricular zone progenitors, you expose them to tritietethimidine to label them, and then you transplant them into P2 animal. This is an animal that is actively producing upper layer cells. You will obtain layer 2-3 cells, and this means that the progenitors that were producing layer 4 neurons are competent to give rise to the layer 2-3 cells. However, if we bring these progenitors back in time and transplant them at E30, that is the moment in which layer 6 neurons are generated, these progenitors of the layer 4 give rise again mainly to upper layer cells, indicating that these cells, these ventricular zone progenitors at E36, are restricted in their cell fate potential and are not able anymore to give rise to layer 6 neurons. These experiments have been interpreted in support of a common progenitor cell that progressively restricts its fate potential. However, these experiments are also compatible with the model of multiple progenitors. Maybe you have to think a bit on this concept. So stop, think, and play again. One of the main drawbacks in studies of the cell fate potential of ventricular zone progenitors has been the lack of specific markers to label neocortical cell types. However, in the last year, numerous specific markers of neocortical neurons have become available. These markers are mostly transcription factors that are involved in the specification of neocortical neurons, and their list is really constantly growing. The availability of this marker was fundamental for a second landmark work supporting the model of a common progenitor. In this study, individual progenitors from the mice embryonic neocortex were isolated in vitro, and their daughter cells were followed with time-lapse video microscopy. In this slide, you can see the main markers that were used in this study. Here you see reelin, that is among the first cells that are produced during neurogenesis are reelin positive. Actually, in vivo, they are not produced by the cortical ventricular zone, they are produced elsewhere, but maybe in vitro was different. We don't yet know, actually. However, then you can see ER81 that is expressed mainly by a subset of layer 5 neurons, FOXP2 that is expressed by layer 6 neurons, TLA4 that is expressed by layer 5 and layer 6 neuron, and COX1 that is highly expressed and specifically expressed by upper layer neurons 2, 3 and 4. Well, cells were isolated from the ventricular zone at E10, that is before the onset of neurogenesis. As you can see, layer-specific cell types are generated in the correct order in vitro. Here the days in vitro are marked. As you can see, first come the reelin and TLA4 cells, and then the production of these cells decline while it begins. We begin to see COX1 positive cells that is a marker of the upper layers. Actually, it is to note that upper layer cells are produced with very low efficiency. These cells are the more abundant cell types in the adult cortex in vivo, while here very few cells are produced as compared with other cell types. By using time-lapse video microscopy, the authors could draw the lineage of individual progenitors. As you can see, these lineage trees are asymmetric, and with progenitors giving rise sequentially to different cell types, and initial division give rise to reelin positive cells, then or FOXP2 positive cells, and then the ER81 or the TLA4 cells are produced, and according to what happens in vivo, these cells are produced in late division. Interestingly, however, these authors did not show the progenitors giving rise to both upper and lower layer neurons. Actually, in this figure, there are only two lineages of progenitors that contain COX2 positive cells, and these are from progenitors isolated from the A12 neocortex, so are later progenitors. Thus, this study provides evidence that a single cortical progenitor can give rise to multiple cell time, but at the same time it do not clarify whether the lower layers 5 and 6 or the upper layers 2, 3 and 4 have a common progenitor. Okay, now it's up to you to discover the ends, for the moment, of this story by reading these two papers. That one is from Science in 2012, the other is from Neurons in 2013. These are both very good journals. However, I want you to read critically this paper, because both have their limitation. They maybe there are different ways in which you can interpret their results. So I want you to be as much critic as possible. Okay. And so no, I don't want to go into excessive details. You can find here two slides that can be introductory, or however they gives you some images, additional images, supplementary of the supplementary images. And anyway, good reading. We will listen again in a chat. Bye bye.